ecancertv would definitely like to congratulate you on being the AACR Distinguished Lectureship awardee, and you have chosen to talk about a topic that's very close to your heart, molecular evolution, specifically here molecular evolution under neoadjuvant chemotherapy. So I'm going to ask you all about this. What is the context of this in breast cancer, and what are the exciting areas that you've been looking into and you want to tell the world about?
Yeah, no, I think that evolution is extremely important in breast cancer progression and what is ongoing when you are on any kind of chemotherapy, for example. And of course, since we know that breast cancer is so many heterogeneous diseases, and we were among the pioneering in classify them into these 5 subtypes, we have been asking the question, is the evolutionary path different in the different subgroups?
And is it?
It looks like it definitely is, and how does that alter during the treatment? And can you sort of move from one phenotype to another phenotype?
And can you predict this evolution, though?
Well, not today, but that's of course some of the aim that we really would like to predict the evolution. And I think that having one snapshot of the sample, you cannot predict. So then when you have 3 samples, for example, when the patient are under treatment, then you may be more able to predict than just having one snapshot.
Now, we tend to initially categorize patients according to the initial biopsy testing, but now you're proposing serial testing. What's happening during the period of neoadjuvant treatment that you're particularly addressing today?
No, because one of our aim is also to do classification and follow the molecular evolution at many different levels, not only at the DNA level, but the DNA copy number, the sequence alterations, the methylation, but then also on the microRNA level, the mRNA level, protein, and the metabolic level. So that's what we have been doing, to do at 6 different levels to put that together. And that can tell us more about the dynamics between the different subclones and what is changing during this treatment.
Now, is it the presence of different clones, or is it the ambience created by the therapy that's the main driver of this evolution?
Well, I think it's without any treatment, the tumor will evolve anyway, but I think it's on a larger stress, and you have more selection when you have, of course, a stress on the tumor cells, and so you eradicate some of the most proliferating one. That gives room to the less proliferating that may come further in the next stage. So then, of course, we would like to see if you change therapy during this course, can you eradicate the next subset of clones that you didn't eradicate in the first round.
And part of the success of doing this could well be in the sheer mathematics of what you're doing, couldn't
exactly. it? So we're working close with mathematical modeling to really model this evolution of the tumor cells and the cooperation between the different clones.
Could you hypothesize for me a sort of successful scenario where you do check upon the genetic composition initially, then you follow it and then you change? What kind of thing would you like to see happen with different therapies evolving with the clonal evolution of the cancer?
No, I would say, first of all, if we know about sort of some signatures that can highlight which way the evolutionary route is going, and then if we can transfer that to, for example, in situ technology. So you can do it in situ and look by cell, are they close by? And then you can do that on the sections as you go along. And then you can immediately follow that patient, take the biopsy, look at the section, look at the clonal composition, and then change the therapy, because then you can see which clone has been dying and which is still left, and can you target that one. And I think that's sort of the scenario that I hope to see. And in addition to that, of course, if we can follow circulating tumor DNA at the same time, you have 2 really highly powerful technologies that you can follow and do changes in the patient much, much earlier.
So you need both the biopsies and the tumor DNA, circulating tumor DNA, do you?
I think that would be absolutely the best to have both at the same time.
But you could monitor more frequently with the circulating
You
DNA.
could do, and of course that's non-invasive, so you can do that more frequently, and then you can take the tumor biopsy. But for the time being, I think we know what happens actually also in the tumor itself.
What do you think doctors should make of this technology and this science and this mathematics right now? Is it a distant hope to be able to get ahead of the game and halt the cancer despite the proliferating number of subspecies that are happening there?
Yeah, I think it's a hope that it's, because we still have some work to do to really find the right profile and the right patients where we can start to intervene. But I think we need to start to do some trials in this to really follow it and to see how much benefit it will be to the patient, and be allowed to change the therapy more frequently than we do.
And what kinds of innovative intervention could be used, do you think?
Well, first of all, I think we need to do more combination therapy, because one single target is not, so we need to have more targets at the same time. And because then we have to rule out the toxicity, and then maybe a new combination in the next round.
And the impact of things like radiotherapy on all of this, does that have any impact at all?
I think it will. We know too little about that for the time being, how much of the radiation is impacting the clonal evolution. But that's another study that we really would like to take on.
Of course, the trade-off between more immunological approaches and more chemotherapy, more chemical approaches to anti-tumor, how does that play?
Well, I think, for example, if you can monitor also, because when you're really looking in situ, and that's the advantage of having the biopsy, you also see the infiltrating cells, you see the macrophages and the TILs that are coming in there, and you can see how they act. And if you can stain which type of TILs are there, you can also use that as a marker for, are you going to do now immunotherapy in this ones?
A very exciting field, but what is the take-home message for practical cancer doctors who have patients right now?
Well, I think for the practical, from my view, is collect samples, biopsy, and we need to do investigation to do that.